So this is going to be my video for my early plant development quiz for my botany class. So first question is to compare and contrast monocot and dicot seeds using diagrams. And here is my monocot seed diagram. And I'm going to start off by just listing off what I have here and explaining and giving a short description of their functions. Okay, we have the endosperm. This basically serves as food storage for the seed. And then we have the seed coat, which is just this outer skin of the seed to basically protect it. And then we have the scutellum right here. This right here is said to help the embryo absorb the endosperm. Basically helps with energy. Helps basically distribute energy to the embryo. And speaking of the embryo, this is going to be that big green area right here. And then there's some more little parts of that. And from here, this is an enlarged picture of the embryo. We have the coleoptile. Forgot how to pronounce that. But this right here is basically, this protects the plumule, and the plumule is going to be when the plant finally sprouts out of the seed. This is going to be the first leaf in the monocot. Monocots only have one, I think, when they sprout out. And then from there, there's the mesocotyl. The mesocotyl is basically the middle part of the cotyledon. And this is basically just separates the plumule and the radicle. And the radicle here, this will be the first root to sprout out of the seed. And then from there we have the coleorhiza, yeah. And this is basically just protects the radicle in the seed. So that's my monocot diagram, and here's my dicot diagram. From here we have the endosperm, and like I've said before, and like basically the function is the same in this one than it is in the monocot seed. And then we have the seed coat, again the same as what I said in the monocot seed diagram. Then we have the embryo. This one's going to be a little bit different. And here is the embryo but enlarged. So right here we have the foliage leaves. These are basically going to be the two first leaves that sprout out of the seed when it becomes a plant. And then we have the epicotyl right here, which is the upper part, the upper part of the cotyledon. And then we have the hydrocotyl, which is the lower part of the cotyledon. Basically, this will just become the root axis of the plant when it sprouts out. So yeah, that's what I got for my dicot seed. And to compare the two, they basically both have endosperm. They both have embryos. They both have seed coats. And yeah, I think that's about. They both have seed coats. Yeah, I think that's about it for the similarities. Oh, they both start out as seeds. And they both sprout out roots and stems from their seeds. So yeah, those are some similarities that I can think of. And then some differences. Okay, so basically the main differences are going to be in the embryos, which for the monocot seed there's quite a bit of little separation of the embryos, and then we have the whole scutellum right here, which I don't think dicot seeds have, but they have the scutellum right here, which basically helps with energy or food consumption of the embryo. And we have 1, 2, 3, 4, 5 different little parts that I just listed right here. When in the dicot, I only had about 3. Yeah, so this one isn't as complicated as what it is on the monocot seed. So I would say that the biggest difference between these two would be the embryos. So that's my contrast of the dicot and monocot seeds. Now, for the second question, I need to compare and contrast epigeous germination and hypogeous germination. And I have 2 diagrams for that also. For epigeous germination, basically the seed gets, or yeah, basically the hypocotyl, basically the cotyledon gets dragged out of the soil and brought above the surface, basically sprouting out into the whole plant, where there's those, yeah, basically the cotyledon gets dragged to the surface. And yeah, this is my diagram for the epigeous germination. The big definition for this would be the cotyledon being taken out, basically just pulled out of the ground, and helps with the sprouting of the plant even more. And then the hypogeous germination I have right here. Here, the cotyledon stays in the ground. It doesn't get pulled out. If anything, it just sprouts out a stem and roots, and just stays in the same place while everything else is growing from it. Yeah. So basically, the biggest difference between the two is where the cotyledon stays during the process of the plant germinating. And the similarities would be they're both the hypocotyl, basically they both have hypocotyls and they both have epicotyls, obviously, but well, I can list a little bit more differences. Actually, the hypocotyl basically comes out of the ground in the epigeous germination. In the hypogeous germination, it basically stays in the ground and stays and becomes the roots. In the epigeous germination, it seems to be part of the stem, where the epicotyl comes out of the cotyledon, which makes sense, the upper part of the cotyledon. But here, the epicotyl, it becomes the stem, and the hypocotyl becomes the roots. Similarities is they both have roots, and they both have stems, and they both have seeds, and they both involve the ground somehow. But yeah, that's what I can think of right now for time, anyway. And then for the third question, I have explain embryogenesis. Embryogenesis, my diagram's kind of big. The other part is it's on another sheet of paper. But basically, with embryogenesis, we start out in the zygote stage, which starts out with the fertilized egg. With that fertilized egg, through fertilization, it basically just starts to cell division, which would basically make the apical cells and the basal cells in that process. And these also start dividing and becoming, basically multiplying and making their structures larger. As for with the apical cells, basically they start replicating and they start bunching up together to make basically a big globe right here, which is called the globular stage. In the globular stage, the apical cells divide and they all link up and they all group together and make this big globe on top. And the apical cells, the basal cells basically start elongating, becoming longer, and from there, and yeah, they end up replicating, becoming longer, and becoming suspensors. Well, a suspensor in this case. And this is just a midpoint between the globular stage and the heart-shaped stage. In the heart-shaped stage, this is where the apical cells divide and they start forming into a heart shape, where these 2 points right here are going to become the cotyledons. And here's another show of the suspensor right here from the basal cells. And here is the torpedo stage. The torpedo stage, basically the cotyledons are fully formed. They're fully formed. And then this is where we also have when the shoot this is where the shoot apical meristem will start to develop also, and where the root apical meristem will develop. And this is going to be towards the middle, on the bottom part of the apical cells. So that's the torpedo stage right here. And then I have the mature embryo stage on this other piece of paper because it's so big. So here it's basically the final stage of an embryo, where the epidermis starts forming, which is the skin of the embryo. And this is where vascular tissue forms. Here's the here's the shoot apical meristem. The root apical meristem will be around here somewhere. But from right here, there's ground tissue, and then these big old boys are the cotyledons. They got huge. And we have our basal cells still, that in a this in a suspensor. But yeah, and this stage is basically when vascular tissue, ground tissue, the epidermis, well, the epidermis forms the ground tissue and vascular tissue, and apical meristems. They basically start cellular division. They start dividing and getting bigger. Well, not getting bigger, but multiplying cells to get bigger. Yeah, there we go. That's a better word for it. But yeah, that is my answer for the third question with my diagram. I don't know. I have a flipped camera. It's kind of hard, but it's kind of huge. This is the mature. This is the mature stage of the mature embryo stage, and here are the other ones, just to show them off again. And that's my entry. Thank you. Everything that I got, all the information from this video is from the lecture on early plant development. So yeah, thank you.